So before genetics, of course, we did have some idea of the pathophysiology of Parkinson's disease. And the main candidates, which predominantly arose from investigation of post-mortem samples, were mitochondrial dysfunction, oxidative stress, inflammation, calcium dysregulation. And these themes have been retained, in fact, supported by the identification of genes relevant to these particular areas as causes of familial Parkinson's disease. But other pathways within the pathophysiology of PD have been identified, and I'm going to focus in particular on those. This slide shows the calendar of gene identification in Parkinsonism. Not all of these gene mutations cause idiopathic Parkinson's disease, but most are related to Parkinsonism. And they can be divided into those that cause familial Parkinson's disease that are relatively rare, that you see in the top left here, and those that are risk genes that are much more common but have less of an influence on the disease itself. And in the middle sit two genes, in particular LRRK2 and GBA, which I'll talk about a little bit more. These are variably penetrant in some cases, like LRRK2, a mixture of autosomal dominant and recessive, the recessive like PINK1, PARK2, and DJ1. Now, in terms of pathways, two common pathways that encompass many of these genes that are associated with Parkinsonism are either mitochondrial or lysosomal, endosomal, or autophagy. In terms of the mitochondrial genes associated with familial Parkinson's disease, Parkin, PINK1, DJ1, and FBOX7 are the most common. And interestingly, they are associated with the mitophagy pathway. This is the means by which mitochondria are turned over, so defective mitochondria are autophaged by a pathway that is triggered by PINK1 and Parkin, and also contributed to by FBOX7. So these genes play an important role in the turnover of defective mitochondria, and it's thought that mutations in these lead to a buildup of abnormal mitochondria that then might promote not only abnormalities of oxidative stress, but also oxidative phosphorylation. Inevitably, these have become a target for therapeutic interventions, and a number of studies have attempted to improve mitochondrial function and delay Parkinson's disease. These are the ones that have been introduced most recently and are currently under trial. So we await the outcome of these studies to see if they can improve mitochondrial function and thereby delay the progression of Parkinson's disease. It's the second pathway, is the conjunction of the lysosomal, endosomal, and autophagy pathways that I think have attracted most attention. I'm going to focus on a little bit more. In particular, I'm going to talk about GBA as the archetypal lysosomal genetic association of Parkinson's disease. So this, as I mentioned before, sits in the middle. So these, both LRRK2 and GBA, are association genes, if you like. LRRK2 perhaps a little bit more in terms of its dominant inheritance pattern, but with variable penetrance. GBA is probably best considered as a risk factor for Parkinson's disease. Mutations of the GBA gene, or glucocerebrosidase, are familiar to those that know the ceramide pathway, where mutations of the gene here, marked with an arrow, are a cause of Gaucher disease, which is an autosomal recessive disorder. And it was the identification of an increased number of individuals with Gaucher disease and Parkinson's disease that led to the association of the GBA gene with Parkinson's disease itself. And I'm going to tell you a little bit more about that shortly. Just in terms of the enzyme glucocerebrosidase, this is a protein enzyme which is encoded by nuclear genes, which is sent to the ER and Golgi for post-translational modification, and then transported via LIMP2 receptors to the lysosome, and plays an important role in the destruction of proteins and other parts of the cell that are destined for recycling. Now, GBA and Parkinson's disease. So an interesting observation is that there doesn't seem to be a significant dose effect. So 1 or 2 mutations of the gene, that's to say GD or apparently asymptomatic carriers, have a similar risk of Parkinson's disease. And up to 30% of them will develop the disease by age 80. If we look at it another way, our Parkinson patients in the clinic, about 10 to 15% and 25% in the Ashkenazi population have GBA mutations. These mutations are also associated with dementia with Lewy bodies. The 2 commonest mutations are the N370S and the L444P. Interestingly, another common one, the E326K, occurs in Parkinson's disease, but not Gaucher disease. In terms of clinical features, there's very little difference between those with and those without GBA mutations. However, a GBA mutation will confer a slightly earlier onset of the disease, some more cognitive impairment, and perhaps faster progression. And this is related to the type of mutation. So here you'll see a graph that shows the most severe mutations here at the top that show more rapid progression than the most mild mutations or those with no mutations in terms of the development of dementia. The pathology, imaging, and pharmacology are virtually identical between those with and without GBA mutations. And interestingly, those that are GBA mutation carriers without Parkinson's disease may exhibit prodromal features of Parkinson's disease, including impaired olfaction, subtle motor deficits, cognitive dysfunction, REM behavior disorder, depression, constipation. So it's interesting that asymptomatic carriers develop this before the diagnosis of Parkinson's disease. And also interestingly, a recent study has demonstrated that GBA mutation carriers without Parkinson's disease have evidence of microglial activation in the brain, particularly in the region of the substantia nigra. And this occurs without the loss of dopaminergic neurons as defined by PET. Let's just turn briefly to the biochemistry. So those with GBA mutations and Parkinson's disease, not surprisingly, have a reduction in the enzyme activity. But what is more surprising and very interesting is that even those Parkinson patients without a mutation, they too have a defect, although not severe, in the activity of this same enzyme. And this association between the enzyme, the GBA enzyme here called GCase, and alpha-synuclein is very important to the basics of the pathophysiology of GBA-related Parkinson's disease. Any reduction in the enzyme activity will lead to an increased accumulation of synuclein. And interestingly, any increase in synuclein will also lead to a reduction in GCase activity. So here you have the potential to set up a self-amplifying cycle that lies at the core of probably GBA-related pathology. Interestingly, the presence of a GBA mutation in an individual who is developing Parkinson's disease or Lewy body-related pathology will lead to an acceleration of the spread of that pathology. Here, a study showed that the injection of preformed fibrils of alpha-synuclein into GBA mutant mice led to a significant increase and acceleration of the spread of alpha-synuclein through the mutant mouse brain. So in summary, the GBA mutation is a common risk factor, indeed the commonest risk factor for Parkinson's disease, elevating an individual's risk by 20 to 30-fold. And it's present in up to 10 to 15% of our Parkinson patients. Importantly, not everyone with a mutation will develop Parkinson's disease. Indeed, only 30% by some studies. They cause prodromal symptoms, microglial activation, increased synuclein levels, and the spread of synuclein, and clinically, earlier onset and more cognitive dysfunction with faster progression. So this brings us to the issue of therapeutic intervention. and how we may leverage our understanding of this pathway in terms of slowing down Parkinson's disease related to GBA mutations, but perhaps also unrelated to GBA mutations, i.e. idiopathic Parkinson's disease, given that those patients too have a reduction of this same enzyme activity. So here's a pathway at the bottom of which is the GBA mutation and reduced enzyme activity. Now, a reduction in enzyme activity will lead to an increase in substrate levels of, for instance, glucosylceramide. And this is seen, for instance, as Gaucher disease, where glucosylceramide is accumulated in cells of the liver, spleen, bone marrow, etc. So one strategy was to reduce the buildup of substrate on the assumption that this was contributing to Parkinson's disease pathology. And a recent trial in phase 2 has come to completion using venglustat as an inhibitor of glucoceramide synthase. Now, this drug successfully reduced glucosylceramide levels, but failed to reach its endpoint of improving Parkinson's disease and has now been terminated. So that was one strategy which has proven unsuccessful. But there are other strategies which perhaps have greater traction. And these include the enzyme chaperones or activators, which will activate the defective enzyme, alternatively to increase GBA gene expression in the patient with a GBA mutation. I want to mention in the last couple of minutes a particular GCase chaperone which has attracted a lot of interest, and this is Ambroxol, a repurposed cough-linctus, which is a powerful GBA chaperone. And this drug has been demonstrated in vitro and in vivo studies to have a wide repertoire of benefit in Parkinson models. So for instance, in fibroblasts from patients with GBA mutations, this drug increases the activity of the enzyme and reduces other abnormalities within the cell, such as ERAD. In flies with GBA mutations, this drug increases brain enzyme activity and protects against neuronal loss and restores the motor function or flight function of the fly. In human iPS, dopaminergic neurons derived from patients with GBA mutations and Parkinson's disease, this drug increases the enzyme activity and reduces alpha-synuclein. In mouse models of GBA, the drug again increases brain GCase and reduces alpha-synuclein levels and spread, and likewise in non-human primates. And perhaps most importantly, based upon this portfolio of in vitro and in vivo drug action, Ambroxol was used in patients with Parkinson's disease in a recent study published last year in JAMA Neurology. Now, this small proof of principle study used patients with and without GBA mutations, but all with Parkinson's disease. And the proof of principle was to determine the efficacy of the drug in reaching the brain and engaging its target. The trial demonstrated that the Ambroxol was safe in this population for a duration of 6 months, and that the CSF concentration of Ambroxol was 11% of that of the blood, indicating very good blood-brain barrier penetration, and was effective in increasing the GCase enzyme protein levels by 35%, and was active in both GBA mutation positive and idiopathic patients. So this drug has a promising future, at least in terms of the next phase in trial, to see if it is capable of improving the outcome of patients with Parkinson's disease. So understanding the genetics of Parkinson's disease has reinforced or directed us to new pathways in the pathogenesis of this disorder, which in turn enables us to target those pathways in order to identify drugs that may reverse or prevent progression and improve outcome for Parkinson patients. And at that point, I'll end. Thank you.
Excellent. Thank you very much for a comprehensive talk in a very short period of time. I think we have time for one question, perhaps. If there are no questions, perhaps one of the very interesting areas at the moment are studies or considerations of studies in the prodromal phase of Parkinson's. As we know that the prodromal phase, which you alluded to, might be 10 or even more years. Some of the non-motor symptoms may start quite early. Do you think there is a role for considering some of trials even in this phase with some of these genetically determined, certainly in these subgroups of patients with genetically determined risk factors?
Yeah, thanks, Annette. So hypothetically, yes, practically a huge challenge. Because even if you take a very enriched population of, let's say, those with GBA mutations and REM behavior sleep disorder, who are much more likely to develop Parkinson's disease, let's say, than idiopathic patients with olfactory loss or other prodromal features, the percentage change of conversion of those individuals to Parkinson's disease, which would inevitably be the endpoint of that type of study to prevent the onset or delay the onset of the disorder, would take an awful long time to undertake. So I suspect that whilst that is extremely attractive, until we have another technique, perhaps imaging, that will provide us with a means to identify some marker that is likely to delay progression, let's say delaying synuclein accumulation, I suspect that clinical trials are much more likely to focus on individuals with a mutation, with Parkinson's disease, and look to see if the drug intervention will slow time to a specific endpoint, which in itself has many difficulties, but nevertheless is practically easier.
Thank you very much.